Francis Ouellette, and I'm proud to be one of the co-chairs of the ISMB 2020 conference in Montreal. And I would like to welcome you there in great numbers so we can enjoy a great scientific conference.
As you finish marking your diaries for ISMB 2020 in Montreal, please welcome to the stage Salvador Capella from ECCB 2020.
Welcome. Welcome, everyone. So I just want to invite you all to the ECCB 2020 in Barcelona, where we expect great science, great venue, and I will say a lot of fun in Barcelona. So let's check out the video. Thank you.
Please welcome now to the stage Torsten Swede, ISMB ECCB 2020 co-chair.
Hello, good morning, everyone. Thanks for joining us this morning, even after the party last night, for our Wednesday keynote. Life is full of surprises. When I agreed to chair the ISMB ECCB 2019, I had no idea that I would have the honor to introduce the Overton Prize winner, Keynote, by a Swiss speaker, by a colleague affiliated with the same institute as myself, the Swiss Institute of Bioinformatics, and that he would be talking about a topic which is very close to my own interests, benchmarking. The Overton Prize is an annual award for an outstanding accomplishment to a scientist in early to mid-career stage, and it's my great pleasure to introduce to you the Overton Prize winner 2019, Christophe Dessimoz. Christophe was selected for the Overton for his renowned contributions and to subsequently manage the OMA resource providing high-quality information on autologous proteins, as well as his rigorous approach to benchmarking, which had major impact on three subfields of computational biology: ontology, inference, sequence alignment, and gene ontology. Had to read the notes from the committee that made this very wise decision. When I was preparing my notes for the introduction, obviously I was asking several colleagues and friends of Christophe for some anecdotes from his former life and some juicy gossip. And I have to tell you, I failed miserably. Christophe is one of these rare people that everybody knows and about whom you only hear good things. Well, you even hear things that are too good to be true. Like somebody told me that he manages to reliably answer all his emails in a timely manner. So if that would be true, Christoph, please tell me how you do that. Not true. So no funny stories in my introduction about Christoph, only praise for an outstanding young scientist and a great colleague. Also, Christoph had an affinity for computers already during his high school times. He decided first to study biology at the ETH in Zurich, with several experimental state visits at the United States, in China, and in Thailand. For his PhD, then, he returned to his initial passion for computer science, and he joined the lab of Gaston Gonnet at the ETH in Zurich, working in the field of comparative genomics. Christoph's first paper introduced the autologous matrix algorithm, which forms the foundation for what we all know today as the OMA database. For his postdoc, he joined Nick Goldman's lab at the EBI, working in large-scale phylogenetic models. During this time, he also developed his interest for benchmarking and the critical assessment of computational models, which we'll hear about more later. In 2013, Christoph moved to the University College of London, and in 2015, he received a professorship award from the Swiss National Science Foundation at the University of Lausanne. So he currently maintains two groups, one at the University College in London and one at the University of Lausanne. This ability to be delocalized, you may also have realized here at the conference, where at least I had the impression that Christoph was present at several COSIs at the same time. Since his project in Gaston Gonnet's lab, Christoph has been a member of the Swiss Institute of Bioinformatics. And this special environment gave him the opportunity to combine cutting-edge science with methods development and to transform it into a service for our community. He stayed involved with the ISCB since working during at the UCL, and now also as a professor in Lausanne. Since about 1 year, Christoph is a member of the board of directors of the ISCB, where he actually replaces me as I stepped down last summer. And it's amazing to see with how much enthusiasm Christoph brings in new ideas into our community here. But he's not only active in the national Swiss bioinformatics community, also internationally, he is somebody taking the initiative. For example, here at ISMB, when he realized that the field of evolution and comparative genomics was losing attention, Christoph took the initiative and revitalized the field, and today it's again a vibrant community as part of ISMB. For his talk, he selected a rather untypical topic, benchmarking. It's an important aspect of any computational method, but it hasn't yet received the attention it deserves in our community. As usual, Christoph decided to do what he thinks is right and not what would be easy or expected. With the Overton Award 2019 for Christophe Dessimoz, the Society recognizes a researcher who gives an excellent role model for the next generation of scientists in our community. Christoph, we very much look forward to your presentation.
Thank you, Karsten. So good morning, everyone. Thank you very much, Torsten, for the very kind introduction, and thank you all for being here. Well, actually, I could almost skip now the introduction. So I'm, in my lab, we are mainly interested in developing methods. So there was mention of OMA, our Orthology method and resource. We also develop methods for phylogenetics, function prediction, and software that works on ontologies and sequence analysis. And then we like to apply this typically in collaboration on a very broad range of topic in a somewhat opportunistic way, and I think this is gives us a chance of making a tour of biology. That's very rewarding. For this presentation, indeed, I thought I would really focus on benchmarking, which is something obviously that we are all confronted with in the course of developing methods. But really, first, and maybe Torsten stole my thunder here. It may be a bit unconventional to have an entire talk about benchmarking. And in fact, those who know me will be really shocked that I don't want to talk about orthology. But although, I will sprinkle a bit of orthology throughout the talk. And maybe those of you who who don't know me just maybe think, well, you know, benchmarking, isn't it the thing that you do once you finish developing your method and you just need to get it published? And of course, you know, this is really misguided, and I hope that you know, our experience here, and as well as maybe some echoes that you might find in your own work, will really convince you that this is not the case. In fact, while I'm saying this, but I'm really by far not the only one to be thinking, and it seems to be really kind of a current issue, and I think this is really like the right forum for having this discussion. I mean, we have people from this community who are extremely active in furthering the cause of benchmarking, be it, you know, as part of you know, encouraging this in the publication record. You may have seen this recent editorial in PLOS Computational Biology. There's an ongoing special issue in genome biology, you know, so with the members of this community. And of course, you also know about all these competitions that are going on, and some of which are based also at ISMB. And so I think this critical assessment series, I mean, obviously CASP and where Thorsten and many others, also from the 3DC, have been active for many, many years, but also many more recent initiatives. And if your area is not yet covered, in fact, there is even literature on how to start new benchmarking efforts, and yeah, so, and also some reflection about specifically like periodic benchmarking. So I feel that you know, this gives us really ample justification to spend a bit of time on this important topic. And so I've selected kind of 3 types of like benchmarking issues that we've encountered in our research, and I think that maybe hopefully will be a bit thought-provoking. So the first one is about dealing with a ground truth that isn't always representative of real data. Okay, and so this is joint work with Adrian Altenhof, Romain Studer, and Marc Romain-Sorachavi. Okay, so promise you a little bit of orthology. I'm really keeping it to the minimum. So imagine like 2 rodents, and they had like a pair of duplicated genes already in their last common ancestor. You know, that could be, for instance, the 2 insulin genes in the rodents. So the pairs of genes that are descended from the same copy in their common ancestor will be referred to as the orthologs. And then we've got some paralogs, which are the copies that result from the earlier duplication, so be it in the same species, or across species. And so that we'll refer to these as the between species ortholog or different species orthologs, whereas the other one are like within species or between species paralogs or within species paralogs. And so one question that had been you know, floating around for some time was really, you know, is there actually a difference in functional conservation between orthologs and paralogs, and in particular, you know, how strong would be this difference? And you know, actually, if you talk to people and you think about all the work that has been done, you know, on model species-based research, it seemed pretty obvious that we would be looking at something like that, where, you know, surely there is a dependency on how distant your species are, but in general, you know, if we correct for that, we would expect orthologs to be more similar than paralogs. So how can we test that? And so, and I should say, so this is referred to as the ortholog conjecture. This is a term that was coined by Nerd et al., including some folks, and Pedja actually, who's with us. Well, yeah, obviously, using the term conjecture also elevates maybe an interesting question to perhaps something that is really worth pursuing. So now, gene ontology, functional annotation can be used now to maybe tackle this question. So I'm sure that most of you are familiar with this, but you know that this is a way to annotate the function of genes. And so here, for instance, I've taken hepatic stellate state activation, and you can see we have here a hierarchy in the ontology, where we can go from specific here to general. Of course, the ontology can be more complicated. We'll have some branches. And so it seems like this is a good source to test this question. And so I mentioned NERD and colleagues, and so they published a paper in 2011 that really came as a big shock to the field. And because what they observed was really very different from what we had expected, in the sense that the paralog, and specifically the same species paralog, seemed to be much more similar than orthologs and different species paralog. So we'd also been working on this question, and so many people were really puzzled. Okay, why is that the case? And so, we can try to maybe think that there's the same species paralogs are in the same cellular environment. Maybe there's something special about really that aspect. But of course, I mean, they and us and many others were trying to find, could there be a confounder? Could there be some problems? And so, obviously, we looked at lots of different possible causes for this, but in the end, really the confounder that really, I think, explained most of this pattern was related to Simpson's paradox. So I think many of you will be familiar with Simpson's paradox, but I think this is such a kind of a powerful concept when we're dealing with large-scale data that I felt it was important to mention it specifically. And Simpson's paradox is mainly known in the context, or as, like, let's say the prototypical example is with the Berkeley 1973 intake. And so at that time, so people noticed that, in 1973, that the uptake rate was much higher, significantly higher in men than in women. And here my circles refer to the number of people involved. And so immediately the questions were, well, okay, there must be some discrimination against women. And actually a lawsuit was filed. And then when they did some analysis, department by department, they noticed, well, actually, that doesn't really seem to be the case. You can see here, that department, women actually have a higher intake rate. Here it's slightly higher. Here it looks to be either a bit lower or higher, but the difference is much smaller. So how can it be that even if within each department there is, there don't seem to be any discrimination, at the end we get such a difference? And yeah, the answer was that if you look at the size of these bubbles, the women were going more selectively to the harder department. You can see many males would select, this large department which has a relatively high intake rate. And so, as a result, when you average all of the data, you may get a very different trend. And so, this is this example, but of course, we are encountering this, everywhere with aggregate data. And so one way to see this is just one illustration, is, you may be able to find a correlation between two variables on pooled data, but when you break this down into single data sets, you get actually possibly really the reverse trend, which is indeed a big problem. So if Simpson's paradox applies to the study of the comparison of orthology and paralogy, we kind of need two ingredients. We need a confounder that may affect the similarity between gene ontology annotation, and we also need this to be quite different in same species paralogs versus the other pairs, right? And so we found actually one that seemed to fit the bill. So, and this was whether the ortholog, the functional similarity involved, well, the pairs of genes that were orthologs or paralog involved gene ontology annotation that were made by the same authors, derived from a paper from the same authors versus different authors. So this is what you get when the average similarity of terms across all of these different categories when they come from the same paper. If they are from different papers, but they have one author in common, you can see there's this major drop. And then if there's no author in common at all, then we can see there's very little similarity. I'm just showing this again. So you can see like high similarity when there's an author in common, and then really quite low. And of course, there's a huge difference between same species paralogs and orthologs when we are looking at the chance of being annotated in the same paper. Think about the fact that people typically run experiments in one species at the time, and it's even actually different communities. In fact, we run the number, it's 50 times more likely for paralogs, same species paralog, to be annotated in the same paper. And so when you correct for this, actually you get a very different picture. And so, okay, the error bars are large, particularly for closely related sequences, but when in the bulk of the data, so which is here represented with the histogram, we can see that actually the orthologs are more similar by a small margin, but this is significant. And so and actually a few other more minor bias could explain this. Actually, I just want to mention two other ones which didn't affect the particular study, but that we found also in our analysis, and I thought that also quite thought-provoking. One is that we observed a huge difference in the average similarity between terms that had been propagated automatically, so inferred automatically, versus terms that are inferred by curators. So, okay, this is a small difference, but it's still, so these are experimentally derived annotations. These are curated ones, and then the uncurated ones tend to be more similar. And in a way, that kind of makes sense because they get copied. And so it's only preferentially like very similar genes and gene products that get annotated automatically with the same function. But this, of course, has a huge impact. If you're doing an analysis comparing, say, non-model species versus model species, the proportion of experimentally derived terms would be hugely different. And so that could be really a treacherous confounder. Another one also I want to highlight is, and which is kind of obvious in hindsight, but we have a lot of these information theoretic measures that depend on like the frequency of terms in many analyses, but this frequency varies enormously from one species to another. And so here is just like a heat map of the frequency, and you can see here number of like the best annotated like model organisms, and with huge difference in the frequency of particular terms. And again, it makes sense, Drosophila is studied for development, maybe the rat for toxicology, and so we would expect these differences. But however, when we're computing, say for instance, the probability of having a term average over the entire database, we are really missing on this potentially very large variation. And, so this is already a few years ago, this study, and since then there's been also many more like follow-up work, and so here I'm just like highlighting a few. I also want to highlight the fact that ISMB has provided also continuously like a nice venue also to keep on working on this and discussing this. There was Tina Begum from Mark's lab last year who gave a brilliant talk, and which is available on YouTube. And this year we had a talk by Moses Tamboulian from Matt Hahn and Peja's lab, who gave a talk in our session yesterday. And in fact, it turns out to be the talk that had generated the most questions. And you can see here like all the queue of people coming to debate this question further. So I think this is probably going to keep us busy in the future as well. Now, if you're interested more about biases and difficulties specifically with the gene ontology and maybe how to control for them and overcome them, we have a nice, I think, book chapter that we wrote with Pascal Gaudet, who's in the Go consortium, is really an expert on all things Go, and particularly how the annotations are made. So I'm moving to challenge number 2. Challenge number 1 was actually the easiest one to deal with. With challenge number 2, we're dealing with an incomplete ground truth. So how can we deal with the fact that our knowledge of biology is typically incomplete? So I'm going back to the gene ontology functional annotation. One thing I forgot to mention in the previous time I showed you this slide is, indeed, for annotations that are derived from experiments, like for instance here from a mutant phenotype, there are some PubMed ID associated with the entries in the gene ontology, which enables us, well, not only to find out who the authors are, but also to look at the evidence. But note, for most species, almost, as a rule, except for a handful of model organisms, everything is propagated electronically, like this IEA. And of course, we're very interested in knowing how accurate function prediction is. And so there's been this great initiative that has had its home also at ISMB, CAFA. And so this, the idea there to verify and to test function annotation is to first release a large set of targets, which are typically poorly annotated, and then ask the community to make good gene ontology prediction on these. And then there's a submission deadline, then experimentally derived annotations are added to the database. In recent years, it was even possible for the CAFA consortium to commission some experiments, to work hand in hand with the expert, for instance, at Uniprots, to generate specifically some, or to not generate, to curate biological function and Go annotation for particular subset of the data. And at the end, there's an analysis that can be done. And so basically, if the predicted annotations overlap well with the annotation that were added afterwards, and which then presumably were not available for the people who did the prediction, then that's great, and otherwise we might have some false negative or false positive. However, a few years back, we noticed that actually, calling it a prediction a false positive if it wasn't annotated during this phase might be a problem, because functions, proteins can have many functions, or at least many aspects of function, and therefore many gene ontology annotation attached to them. And so it's not obvious that not predicting function A necessarily means that predicting function B is a mistake. And we ran a little analysis at that time to see, what if you do this assumption and you wait a bit longer, in how many percentage of the case will you actually then end up with a false positive, so a case where actually there was a good prediction, but we just didn't wait long enough. And so it was quite substantial. I mean, this little experiment, within 5 years, I think we had about 15% of this annotation that would actually turn out to be correct. And so we proposed, we thought, okay, one thing that we noticed at that time is that, we need really to be explicit about having not the lack of an annotation. But that's something that is, and so so that would look like this, right? So where we have a truth set for the positive, but then we only assess the false positive based on a truth set also for the negative, right? And actually, there is this mechanism in the Go, and so this is a qualifier, and it will say this gene product does not have a particular function. So, but the problem with this is that there's a huge imbalance. Okay, there's very, very few not annotation. This is just the annotation that derived through phylogenetic method with the paint method. I'll mention this a little bit more in the next slide. But if you look at the whole database, it's kind of a similar proportion. And when you talk to the annotators, of course, you understand very quickly why they don't like to do not annotation. This is not very. It doesn't seem very rewarding to be spending their time, telling all the things a particular gene product doesn't do. And in fact, sometimes it's harder to tell that something doesn't, because if you want to say that something is not involved in a process in the nucleus, then one experiment may not be sufficient to establish this fact. So there's this huge imbalance, but of course for the field we're starving for this not annotation, and without these, it's really a little bit difficult to move forward. So One thing that we've done recently, maybe a little bit as a proof of concept, but really to encourage also the rest of the community to really be thinking a little bit more about how we could be generating reliable knot annotation in the context of gene ontology, is to try to derive them from phylogenetic trees, gene trees that have been well curated. And so I'm going back to paint. So this is a approach that is used within the Go consortium to annotate entire families. So here we may have, for instance, a family of a DNA mismatch repair gene. And actually, so that, that's a function, an ancestral function. Here we have some fungi, some fungi, and then here the animals. And then maybe, at the root of the animal, this gene is co-opted to be involved in the apoptosis process. And then later, also, we have this somatic hypermutation of immunoglobulin genes that appears kind of here in this case, maybe only within vertebrates. And so what we did is to take this and then kind of turn this onto its head and saying, okay, so maybe that implies that this is not involved in apoptosis and this is not involved in somatic hypermutation of immunoglobulin genes. Now, we can debate whether this is a good assumption, and we can find also certainly some counterexamples, but also discussing this, with our colleagues at the, well, and particularly with Pascal, but also thinking about the fact that, some sort of baseline and alternative kind of assessment would be already a good starting point. We went ahead and tried to retrieve all of these instances. And so if you do that, you get something that is much more balanced in the context of paints, okay? So, but then we realized, actually, well, it sounds easy to be dealing with this negative and positive annotation, like the way I showed it with these 3 circles, but actually there are quite a few tricky implications. And so one thing, and I almost alluded a little bit to that a few seconds ago, is that, I'm showing you a very, again, simplified picture of the ontology, where where you have some terms that are very general. Well, in this case, the root term, so we'd assume that every gene product is involved in a biological process. Of course, some people may disagree with this, and we certainly can find some cases of pseudo genes. But let's say usually when we're looking at, if we're focusing on gene products that are legitimated gene, then this is very general and this is very specific. And so when we are now evaluating our benchmark, of course, we want to differentiate the fact that some predictions are easy to make and some are more difficult. So almost all the benchmarks, or all the benchmarks that I know, will have some sort of weighting to give more weight to a specific prediction. But if we're looking at the negative, now we have things are really very different. I mean, for one thing, we all know that gene ontology terms, they could be propagated to more general terms. If you're part of the fibroblast activation pathway, you're also this implies that you're part of the cell activation pathway. Well, with negative annotation, it's the other way around, right? So if you're not part of cell activation, this means you're definitely not part of fibroblast activation, etc. So propagation is upside down, but also more importantly, the information content is also really asymmetric. So so this has real consequences, because it means that if we take like a very unspecific term, actually it's very relatively easy to predict it in the positive, but it's extremely difficult to predict it in a negative way. And so how can we go around this? And so we kind of need to devise a benchmarking procedure that will balance the two. And so we did this, and we looked at the impact this has on kind of Kappa style, but I'm mentioning Kappa, but this is like nearly all of the papers that you will find in this area, that are based on what we call the closed world assumption, where we assume that not having a particular function, that having a particular function implies not having another one. So when we rerun, so this is basically recapitulating, typical results with the conventional approach. I draw your attention to the 2 baselines here that have been widely used in Kappa. So one is using BLAST, so you blast the closest, you find the closest sequence and propagate the annotation basically, and then your confidence will be how your tolerance in terms of like e-value thresholds. And naive is very naive. Naive is not looking even at the sequence, it's just based on the frequency of the terms in the gene ontology. And one puzzle has been, for some time that naive is kind of doing disturbingly well, right? I mean, so, okay, here it is. So at the end, if you really want a big recall, you will eventually have to call every term, so then it's not doing so well. But it's kind of disturbing to think that in this regime, naive is doing so much better, for instance, than BLAST. Okay. And so now if we do our kind of node-based test, so things look quite different. I wanted also to mention, the thickness here of the line reflect kind of the average information content that was used to estimate, that point. And so actually, we can see here now, naive is not a very good strategy, and I think no one would be here to defend naive. But also, interestingly, I think BLAST, which we know is actually pretty good if you are looking at very closely related sequences, they're actually doing very well, if you are happy with quite a low recall. And I think this is kind of much closer to what we would expect. And so I think, it's really worth exploring a little bit more the use of knot annotation to do this type of estimation and of analysis, and we may get a substantially different picture, which I'm sure will also have some shortcoming. And so this is a process. I really want to stress this. So this is one thing, maybe a little meta discussion now, maybe anticipating some of my conclusion, but it's one thing that I've come to realize in benchmarking is can be very fraught because we sometimes have like some heated discussions. Sometimes our method's doing a bit better, someone else's method is doing a bit better. But I firmly believe that it's kind of a process. It's an area of research, and so we are just doing, a progress one step at a time, learning about, learning from each other. And I'm not claiming by any means that this is the end game. I think, we need to have many more knot annotation also from other sources, and so I hope this is just like one little step forward. Okay, so the last challenge, the missing ground truth. So. Yes, so this is the case where we don't have any ground truth. So in some community that may seem like a really bizarre proposition. I mean, you're maybe thinking, well, just do the experiments. But in our, if you're working with evolution in particular, it's extremely difficult to have, an empirical sense of, how good our predictions are. I mean, we're dealing, for instance, if you think about species tree reconstruction, even with all the data we've got now, there are still some parts that are heavily disputed. Long controversies with back and forth in high-impact journals, and so that seems to be really quite a difficult problem. And so you can imagine if you're now looking at just a single gene and trying to build gene trees or trying to do an alignment, the notion that you will know, okay, this is correct versus this is incorrect, is actually really elusive. So I want to just highlight, this typical pipeline we have in my area, which is, we want to build a tree. So in this case, for instance, that could be a gene tree. We start with the genomes, identify the orthologs. Maybe that already involves some alignment. But here, at this stage, we want to have an alignment of our family. Perhaps retrieve like the good part of that alignment, and then build a tree. All right. So how good are the methods at each step? Okay. So it's not completely obvious, actually. I mean, which, if you think about the number of people who are doing this every day, I mean, it's just sequence alignment alone, isn't it like the kind of the queen discipline of computational biology? And, yet I think we are in a much more tenuous position than we may sometimes like to think. So what are the conventional alignment benchmarks? So of course we could use simulation, and certainly simulation is excellent to weed out bugs. It's excellent to have a complete understanding about where we are doing the mistakes. But obviously all of us who benchmark based on simulation? No, that it's really strongly dependent on the assumption that we put in. And in particular, if we look at our ability to reconstruct, to simulate sequences, I mean, it's quite obvious that we're unable to do anything that resembles like empirical data. And so I think, simulation-based, for instance, alignment benchmark are really quite limited. So really the gold standard has been structure. And those of many people who have much more expertise in this domain than I have. But I think one of the conventional wisdom here is that when you're looking at a typical protein family, structure evolves more slowly than sequence. And so you could find, and this is a really classic paper, and you can see many, for instance, here, these are globin genes that have 20% sequence identity or even lower. So this is, for those of you familiar with sequence alignment, which should be pretty much everybody, this is as good as random, basically, in terms of the percentage identity. You still see many residues that seem to be kind of in the same fold. Okay, so that's great. We can use structure to benchmark sequence, right? However, of course, that's kind of tautological, but that only works for structurally conserved region, right? And so we know, okay, that's in PDB. We still, despite all the progress, we're still very much behind in terms of like resolving protein structure compared to the sequence. But also, I want to draw your attention on like typical benchmarks, which, again, I think these are incredibly important contribution versus not having kind of a baseline. But for instance, in Bali-based, which is kind of highly regarded, it's manually curated, really with experts, there's only 18.8% of the residues that are regarded as really, part of the benchmark for which there's a conserved region, and so the assessment is going to be made. And so that's about 50,000 alignment column, maybe in Bali-based 4. And so my question here is, how many of these 50,000 or so columns contain a gap? Okay. Maybe some of you will venture a guess. Well, the answer is 4. Okay, there's 4 columns that contain a gap. So these are, of course, these are probably correct columns, but are they really representative of the type of alignment that we are doing, days in and days out? Okay, so this is a problem. So as a result, people don't trust alignments. Okay, so there was this paper about 10 years ago, which why would phylogeneticists ignore computerized sequence alignment? I like the idea. The computerized sequence alignment sounds really advanced. This was quite a large survey, about 1,200 papers across different fields. And so again, what's the question? So the question is, how many? What is the proportion of the alignment across all of these papers that were kind of tinkered by hand? And so actually, the proportion is almost 60%, which is really, showing that we haven't really solved the problem of sequence alignment, have we? I mean, the biologists don't believe the alignment that come out of our programs. And okay, so this was in 2009, so maybe, something has happened in the last 10 years. But actually I have, admittedly quite limited the evidence here, but I got a rejection last month. And one of the comments was, the alignment quality seems very problematic when one takes a look at the provided alignment. So I believe perhaps this is, the beliefs in the field has not changed very much. So how can we assess alignment? So what we did, and to maybe contribute to this problem, to contribute a solution to this problem that would be quite different, was to try to assess alignment in terms of the trees that can be built from them. Again, this is, we could discuss also about the shortcomings of this, but the idea is, for instance, here we'd have a pipeline like this, where the only step, in that pipeline that I showed you earlier that varies is the aligner that you're using. And then if you have some ways of evaluating the quality of the trees, for instance, if you start with orthologs, you would expect some congruence with the species tree. Then, if class LW does very well on one on one alignment, perhaps the tree that you reconstruct matches your expectation. That may be a fluke, but if you do that on thousands of gene families, maybe it's telling something about the method. And of course, some people, they don't believe even in the concept of species tree. So we have another test that is looking simply at the number of implied gene duplication. But it's basically the same idea. We basically assessing the quality of the alignment in terms of the resulting trees. And we apply this, to many of these steps along the pipeline. So for instance, for orthology inference, this is very much a community effort. Okay, Adrian Altenhof, my close associate, has been really instrumental in this effort for about 10 years already. In fact, we're celebrating the 10-year anniversary of the quest for orthologs next week in Japan. So it's 10 years. I can be quite precise about this. And so this is a continuous benchmarking effort, and it's really a community effort. We discuss about the criteria. There are many different type of tests, so that we can have like a differentiated picture, also depending on which application of orthology we're looking at. Don't have really time to go too much into detail. I just want to mention also that we are moving now also this benchmark to much more robust infrastructure that Salva is developing called Open eBench, and he's got a talk tomorrow. So if you're interested, you could go there. But so with orthology inference, what we, for instance, noticed, and which really came as a shock to the community, is that graph-based methods, which were typically viewed as like being quick, but quick and dirty, I want to say, compared to tree-based method, they seem actually to be doing rather well on many of the benchmark. And we didn't really see this dichotomy that frankly, everybody in the field were expecting. In fact, just yesterday in the session, someone was referring like the tree-based approaches are like being more precise. And so, this is still something that ongoing. Why is that? And there are different ideas, or maybe I should say conjectures about this problem. Alignment filtering is another one that we looked at. So, if you're building a gene tree, should we remove the dubious sites prior to tree inference? I mean, it sounds, particularly after what I've told you about alignments, people don't trust them. You look at alignment and say, this is obviously wrong. It seems very reasonable to remove the part that are unreliable. No one wants to draw their conclusions from unreliable data. Garbage in, garbage out. And so we looked at a number of methods, quite a few, and then the impact of removing sites on the quality of the trees. And so this is the error in the tree, and so this is worse, and this is better. The baseline is here, basically, if you don't filter anything out. And kind of shockingly, for this particular application, filtering only seems to be worse. And so, in fact, the more you filter, the worse the resulting tree. Which was really also quite a shock and quite a difficult thing to convey to colleagues who are actually doing very good work. And actually, when we took a step back, it's not that this filtering method, and I should say, they are very heavily used. So this is guidance, trimal, G-blocks, you're talking about now thousands of citations. But I want to stress, it's not that they are not doing a good job at removing unreliable columns. Actually, they are doing a good job. And so if we now assess just the alignment accuracy, so obviously, so this we could do in simulation only, so it's kind of limited, but at least it gives us some insights. This is the unfiltered alignment, and you see, well, they are not perfect. They are, some columns are absent. But the proportion of columns that are doing a good job, is relatively high. But indeed, if you filter, you tend to have a higher precision. Of course, you lose columns, so you lose some recall. So actually, these methods are doing what they're supposed to do. But of course, if tree building, for instance, has a very different kind of behavior in terms of like the preference in terms of precision and recall, and specifically, we have kind of our working hypothesis is that it's surprisingly robust actually to errors in the alignment. If you are interested just in building a species tree, it's never a good idea to filter. Which really brings me to an important point, I think, about benchmarking, which is very much context dependent, okay? And so as another illustration of that, that fact, we had a few years ago, like also some back and forth with Des Higgins about a very kind of specific question. So he had a paper claiming that you could get, for very large alignment, you get the best alignment if you use what he called a chain guide tree. So as a guide tree, you would use something that basically adds one sequence at a time. It's not really reflective of, if you've got, let's say, as an extreme case, a group of sequences that are similar on one side of the tree and then another group, you're still better off to add them one at a time. Whereas, the intuition would be, you're better off putting together things, according to the true evolutionary tree. And so it was not very difficult to show that, well, actually, if we are using our benchmarks and if we do simulation, we get actually exactly the opposite. But note, this is now we have to start narrowing a little bit the scope, okay? So this is in simulation and phylogenetic benchmark. And well, I don't know if Fabian or Des are in the audience, but I feel I have to give them the last word on this. So they had like one following paper, and then they kind of doubled down and said, okay, our benchmark score confirm the utility of using this completely chain guide tree. That result has been controversial, but at least for alignment of many sequences from structurally conserved region, the result appears very clear and robust. And so, this is just an illustration how you may have diametrically different conclusion depending on the context. And, I have a lot of trust that, what they've observed is true. But again, it will depend perhaps which part of the alignments you are looking at. So we've got also a book chapter on this called Who Watches the Watchman, if you're interested in multiple sequence alignment specifically. And so I want to finish with just one little piece of unpublished work. So this is about partitioning methods. So some of you will be familiar. I mean, they've become extremely popular in recent years. A tool like Partition Finder has many thousands of citations. So the idea there is, it's kind of an old idea, but now we have computational power, so we can bring it a little bit, you can do that, at a very large scale. And so the idea is, if you have here, this is just a cartoon of an alignment, a concatenated sequence alignment. Typically, we will use the same model and parameters for all the sites and build our tree. But of course, we know different genes may be evolving according to different regime. They might be on the different selection strength, etc. So we need better to have like different subset of the data, kind of partition your data and have different model. And we have the computational power for this, and then we can build a tree. And so maybe we'll look at, well, the conventional thing is if you are working with nucleotide sequence, maybe to look at the three different amino acid, three different codon position. And then perhaps we can combine this in a clever way to find like different subclasses using partitioning method. And we've tested that, and actually it's very time-consuming, but we've tested that in the context again of species tree inference. And so here you have kind of the result where partitioning is giving a better result, and then this is actually worse. And you have sometimes some significantly worse. And so there's three strategies here: partitioning by gene, where there's no optimization at all of the partition. It's just like every gene has its own set of parameters. With Partition Finder, the tool is trying really hard to maximize the Bayesian information criterion. So basically the fits of the data with the model. But now we have many possible models. You can imagine the number of partition is huge. And then there's another partitioning approach based on physicochemical properties. So it doesn't look like, it's not ruinous. These are very small differences. But it doesn't really look like it's helping very much. In fact, rather the contrary, if we look at now statistical significance in a really narrow sense, we could say, it's actually significantly worse in most of the cases. Now, if we look also at CPU time, the picture is even more dramatic. So because you have like orders of magnitudes of difference in compute time, and so we know the distance methods are very quick, but they're not as good. But the gain that you get from partitioning and also actually some mixture models, not included them in the previous figure, is not really justified, at least in our benchmark to the extent that we understand this now. And just that you could see, we're talking about 5 orders of magnitude in terms of compute time difference. So this is very significant. But somehow it's very difficult to go against the common wisdom, the received wisdom of having a more complex model that will somehow fit better our data. But recently, actually, just this year, and I was really pleased to see that there's actually some people who start, questioning this, the notion that you will do a lot of model selection. I mean, model selection, again, is not inherently bad. It's just that, it may be that, there are many contexts where it's not really translating into, like, the improvement that we think it does. Okay. So this brings me to my conclusions. So I think the first point, and I hope that you'll all agree with this, is that if we can't measure what we're doing, we can't improve it. So it should be self-evident that even if our heart lies with algorithm and with method, we want to spend our time, on algorithm and method that will lead to some improvements. So we can't just treat this as an afterthought. I think I really want to highlight, kind of benchmarking as a really legitimate area of research and not just like a mean to an end. And over time we are learning about biases, about how to cope with a number of challenges. And I think it's important that we document this, but also it's also very important that we don't have too much stigma with sometimes making some mistakes. I would not be surprised if some of the results we've obtained still had confounders. In fact, I'm pretty sure that they have some. It's just that we haven't been able to characterize them. And so, if some of us are identifying some shortcomings, I think we're all moving forward. I think benchmarking also requires really a sustained effort. Sometimes we just do it once, but actually, these benchmarks, they have an expiry date. There's new data that come up all the time, new methods. And so, even though sometimes, we just publish maybe the paper with be it as an individual lab or as a community, it's tempting to move forward. I mean, it's really requires a sustained effort, which is a challenge for, let's say compared to contributing a proof, which will, to a theorem that will hopefully, if there's no mistake, will be there for forever. I think one thing that is very important, this is a bit more directed to the younger audience and in this group, is that I think it's very important not to be afraid of questioning long-standing assumption. And we may think, okay, alignment is something that is so ubiquitous and has been going on for so long. I mean, it's kind of shocking, to think that we still know so little about what is a good alignment, at least at the deep evolutionary range, and so for large amounts of divergence. And so we should really be open-minded about this, and also in particular, and for also speaking for someone who's also quite involved in some consortia, don't assume that it's because you have a big consortium with lots of people who've been in the business for a long time that everything has already been figured out. Actually, this is a continuous process, and we have to be really critical about this. Okay, and then finally, I think this is very, very important. I think given a bit of evidence for this, benchmarking is really context dependent, and we're all guilty of thinking, okay, what's the best method, or what's the ranking, but actually this could be really sometimes too reductionist. I mean, I would argue that like even measures like F1 or MCC, like the Matthew correlation coefficient, that try to put together precision and recall, lumped us in this one measure, is also causing quite some disservice. But it's even going beyond that, and also looking at the the context of the benchmark. So I think this is very important. Okay, so these are the conclusion. I hope that we can have a discussion about this in the Q&A session. But before we go there, I want to thank all the people in my lab. So I cannot name all of the people who came through my lab, but I've put my PhD students over the last 10 years, and postdocs, and but also particularly want to thank my advisor. So I'm very pleased that Gaston is in the audience today. So my PhD advisor, Nick Goldman's probably not here, but I also owe him a huge debt of gratitude. I think I'm also extremely fortunate to be in an environment that is really open-minded about this type of question, that values methods, but also values applications of bioinformatics that are really relevant to the broader community. And so I want to highlight, well, certainly my colleagues at UCL, UNIL, and CIB. I mean, I couldn't dream of a better environment to do bioinformatics, but I've singled out 4 people, I think, that have made a particular contribution. So Mark, I think, who's also here, Max Telford, my colleague on the biological application at UCL, Christine Orengo, and David Jones. I mean, they've been really very active in the CAFA community, and the thought that we sometimes were 3 of us, on the same floor discussing, some aspect about protein function prediction has been incredibly valuable. And then finally, I think we have I'm involved in 2 really wonderful communities. So the Quest for Orthodox community, which, we are competing at some level, but at the same time, we're really, I think, collaborating, and we're open also to newcomers. And I think at the end of the day, we are meeting with people who are obsessed about the same minute details is something that is extremely rewarding. I mean, at some level, we are doing this at ISMB in general, but I think within COSI and then within these sub-communities, this can be extremely rewarding over time. Okay, and I thank the founder and all of you for waking up early this morning. Thank you.
Thank you, Christophe. A very inspiring presentation. We have time for questions. If you use, please, the microphones in the corridors, and while you're lining up, maybe I get started with... You mentioned the area where there's no ground truth, and you had kind of substitute data coming from somewhere else, but there are sometimes situations where you can't get substitute ground truths, and then communities resolve to consensus as being the point you compare against. What's your take on that?
Yeah, so I think consensus is a very appealing idea also to anyone, actually. We tend to agree with the majority. It's very compelling to tend to agree with the majority. I see this also when we talk with biologists, the idea that you're testing multiple methods and they all give you the same answer is really comforting, and certainly consistency is a prerequisite. But I think, yeah, of course, these alone, you could be really consistently wrong. And so I think if you only have consistency, I think, it's quite intuitive that things can go wrong also. So I think there's no substitute to at least finding some surrogate that will be outside of your methods, and that will give a bit of a more absolute measure of how things are, yeah.
Please.
Thank you, Christophe, for a good talk. Benchmarking, of course, we know, and actually for raising the awareness of benchmarking. It's a very heterogeneous and difficult problem. So one of my questions, because you featured a lot of our work, I think it's not such a bad light as it might seem, is the dangers of overcorrection. So this is in particular for, of course, in some cases, even when we do benchmark things, we know that there are problems with this, and those are stated, but we can't do better. In other cases, we might be overcorrecting things, and in particular for the ortholog conjecture, what might seem such as a bias, for example, that some proteins are more studied in human, other proteins are more studied in mice. Some other things are not biased, for example, that more genes are olfactory in mouse than maybe in human. So once we do the corrections and multiple successive corrections, we might end up, as in your graphs, with flat curves or functional similarity based on sequence similarity. That looks to me as maybe there's no evolution. Sequence identity doesn't determine function. So where is that balance between correction? How do we strike the right thing in light of all the possibilities?
Yeah, so thank you also for all of your work. I mean, it's wonderful to have had someone to argue with that is also so constructive. So I think in this case, it really, again, goes back to the question about what is the purpose and what is it the claim that we are trying to make. And so I think, if we try to make a claim about like how biology works in general, then of course we should be very worried about the annotation biases. On the other hand, and we want to normalize this, on the other hand, if we want to characterize the differences, for instance, among different annotators, then of course we would not want to normalize this. So I think, again, that this goes to, we can't just say in general, normalization is good, normalization is bad. I think it's very context dependent, and we have to be very precise about this.
Yeah, I agree with that.
Yeah.
Can we have the micro on the right side?
Never mind.
No, it's working. Now it's working.
Oh, okay. See? Got offended. Okay, thanks for a great talk, Christophe. So just a comment really about bias. So one interesting finding about bias is that, especially when you go to the supposedly more reliable experimental annotations, and something we kind of looked at was, and that actually was driven by CAFA, because when we ran CAFA, we found, oh wow, there's a lot of bias going on. And it surprised us because probably not you, but this was the first time we encountered it. And we drilled a bit deeper, we found that bias is driven largely by lab budgets. So there are a few labs with large budgets that do experimental annotations, high throughput, and that drives the bias. And that's a good thing, but we have to be very much aware that the human factor here is incredibly larger than we thought it is. And a nice shout out maybe for EBI, who are now correcting for it by labeling all these high throughput experiments as such, so that people would know how to address them. Of course, I'm not saying we should throw out the baby with the bathwater. Those should be there. So that's yet another, kind of the human in the fold that affects how we view the world and science in general.
Thank you, Ido.
Yeah. Christophe, really wonderful presentation. I have a question, or I would like to know your view on this. We're moving from hundreds of sequences when we do alignment to 1,000, even to 10,000 or millions. And we don't know exactly how to generate those alignments. And we don't know really how to generate a benchmarking reference data set to do that. So how do you foresee that challenge?
So, I'm going to mention some unpublished work now in this Q&A, very briefly. But we have evidence that the aligners, they don't really profit very much from additional sequences. If you're building trees, actually, even though it's a very difficult problem, adding more sequences, well, it's kind of NP complete, and so you would think, it's not obvious that adding more sequences helps. Actually, generally, it breaks small branches, and it really helps. We've got some evidence that for the aligners, they indeed don't deal very well with these very large data sets, and they don't profit from this additional information, which in principle, could be helpful, but it's just computationally very difficult. And so how do you do that? Well, again, we could discuss about some specific strategies, but I would concentrate on very large alignment and look at the consistency of the alignment that are produced with some, some other aspects that we otherwise know about these sequences, perhaps, but again, we should not, probably, it's not the right moment now to discuss about experimental design, but for instance, you could be looking at, if there's a particular active site, is it aligned? But then again, what will it tell you about the non-active site part? So, this is a, it's a difficult problem. It's actually, we have to sit down. Perhaps we should write a grant and then really, think about this in earnest. It's not something that, it's a legitimate research area. That's what I was trying to say. Thanks, Salva.
So can I interrupt for a second? So as you know, we're streaming the keynote online, and we have a question from India, just in the immediate. And Prash is asking, thank you for your interesting talk, Christophe. How would we see alignment benchmarks for simulating metagenomes in the future, and where would you see the potential challenges and trade-offs for that?
I think we're again, back to a similar question as that of Salva. I think, I don't pretend that I can come up with benchmarks just like this. And so I think, indeed, metagenomics is a huge challenge now, because it's changing very much the type of data that we have, the amount of data that we have. It's clearly incredibly valuable. And so we will need to think of ways to benchmark this. And in fact, there are some people, there's the whole community on the metagenomics has been also quite active, also tackling some of these challenges. But I don't pretend that I will know the answer, just so quickly. Yeah.
Thank you. Gaston, please.
Hi, Christophe. Thank you very much for the talk. It was great. I loved your one of the conclusions about if you can't measure, you can't improve it. And I would like to quote Lord Kelvin again. You know that's one of my favorite quotes. If you can't measure it, you are not doing science.
Okay, I think you remained true to your let's say straightforward nature. I would not dare to make such a claim, Gaston. But
it's not me. It's Lord Kelvin.
Okay. I think there's many areas that are really worthwhile, and so I'm merely trying to elevate benchmarking to the same level as the other parts of benchmarking. There are many aspects that are really important.
Okay, Carsten, last question goes to you.
Thank you. Also, thank you, Christophe, for an excellent talk. You focused very much on the scientific challenges in benchmarking. I also see a lot of practical challenges in benchmarking, in particular when you use advanced machine learning models or when you move more to genomics and medicine. Very fundamental problems arise there. For example, you may not have access to the training data set of all the different models you want to compare. They may be commercial. Even the code for retraining them may be commercial or non-available. Or you may not have the computational resources to retrain these models that have been trained by someone else. So there are very many practical and very fundamental challenges to benchmarking there. I wanted to ask you about your vision, how to solve these practical problems in benchmarking besides the scientific ones.
Okay, well, thank you very much, Carsten. I think, so you're absolutely right. And so again, this is why it's a lot of these benchmarking efforts are really ongoing effort. I mean, some of them are, let's say with CASP, it's 30 years, and then still not everything has been figured out. There's still some room for improvement. And then I think with this emerging area, we are still very much at the beginnings. And I think if we accept first that this is really something that is important, that is not just an afterthought, then I think we can also, as a community, require some norm. Saying, unless you've done this type of benchmarking, we're not even going to, to go into details about, whether the method is performing well or not. You've got to first show this on say a broad set of data. And same thing, can you make your code available? Code available is good for reuse, but we have to perhaps indeed in some case make sure that the training sets are available and disclosed, because these are incredibly important. I mean, particularly thinking about things like deep learning, where you have you may have all of the training data that is encoded in some parameters of your network. If you don't know the training data, it's extremely difficult to know what you came back, how these arose and how we can do progress. So I entirely agree with what you say. Again, I'm afraid I don't have really the answers yet, but I'm very pleased, that, well, particularly I think in Switzerland, the ISCB, we have many people really interested in this. I think also in this community, this is really, I think as a community with ISCB, we can really make a difference. Also when we discuss with our collaborators in other area of biology to really highlight the importance of this, and then hopefully that maybe even clinical scientists will also see this benefit. Even though that's maybe quite far removed, but it's really essential. So I completely agree with you.
Please join me in thanking Christophe for an outstanding keynote, and it's my great pleasure to hand you over the Overton Award 2019.
Well, thank you, Torsten. Thank you, everybody.
Thank you. Yours.
Thank you so much.
Vortrag. So great presentation. I loved it.